Welcome to CSEC Biology Live with Mr. Wilson. It's another Friday evening, and we want to get the show up going and, of course, presenting the content that you have so come used to getting. It's overcast, and in some cases, it's really wet on the outside. But despite that, we have to get going. Now, you can join us on a Friday or on a Sunday. We have the Biology HSB Live at 5 p.m. And we are reaching out to all those students who are doing biology across the Caribbean. If you are doing human and social biology, we are also catering for you. Now, last Sunday, we looked at photosynthesis, and a lot of persons message us to say that they were unable to be a part of the group of persons to be a part of the show. Hence, we had moved on and they are left behind. For that reason, we are going to be doing just a little of what we did last week, Sunday, today, and we are going to be adding a little more to what we did then. And when we're asking you at this time to just invite your friends on over, those persons who are supposed to be on the live, there are a lot of persons who said that they wanted to be here. So you could just remind them that it's time for the live, and we're going to be looking at photosynthesis today. We want to bring a wrap to that today, and Sunday, we're going to be looking at circulation, and next week, Friday, we're going to be looking at plant nutrients. So please join me here on CSET Biology. Please join me here on CSET Biology Live with Mr. Wilson. All right, so what better way to ask? Creek is on with us. Creek, I'm going to be asking the first question. What's photosynthesis to you? You can type in the chat. What's photosynthesis to you? Tell me what it means to you. All right, there are many definitions out there for photosynthesis, some which are true, some which are close to being true, and some which are way off. So I'm asking a Creek Williams, good evening, sir, if he could share with us what is photosynthesis. I will give him a few seconds to get going, and then we will say what we think it is. Creek, are you there? Nudge some of your friends for me. All right, good. We got to go. Now, photosynthesis is the process by which plants and some bacteria would be able to make food for themselves using simple inorganic substances to make more organic substance. Now, the inorganic substance that they would be using would include water and carbon dioxide. We did that last week to make complex substances such as, for immediately, they would be making glucose. But that could even go as far as to make carbohydrate, to make protein, to make, of course, lipid and vitamin. So, of course, remember, photosynthesis is simply a process in which plants and some bacteria uses, of course, inorganic substance to make organic substance. And we did give some examples here of inorganic substances. Now, another thing that we needed to know, or that we mentioned last week, was really the three types of nutrition that the syllabus wants us to look at. And then we spoke about autotrophic nutrition, we spoke about heterotrophic nutrition, and we said that heterotrophic nutrition is a type of nutrition in which the organism, which is the autotroph, depends on another organism for food. Now that organism, the heterotroph could be a herbivore, it could be a omnivore, or could even be a carnivore. So what it's going to eat is going to, of course, be dependent on its nutritional type. Then we spoke about the other type which the syllabus looks at, which is saprophytic nutrition. I would say that saprophytes are really feeding on dead and decaying matter. Now, we looked at heterotroph in a bigger spectrum where we spoke about heterotrophic organisms being, we at that point we put saprophytic organism under the group heterotroph. We had placed parasitic organism under that group as well. And we also placed holozoic organism under that group. And I'm going to quickly go through these so that those persons who were not here could catch on so we could move on with the lesson. We said that the heterotrophic organisms that are holozoic, they have a 5-step nutritional process. And we spoke about ingestion just to get the food into the mouth. Then we spoke about digestion to break down the food. Then we spoke about absorption to get that food into the bloodstream. Then we spoke of assimilation. And after assimilation, we made mention of excretion. Is that the 5? So let's go again. Ingestion, digestion, absorption, assimilation, and then excretion. So those are for the holozoic organism, and holozoic organism would be like us, human, right? And of course, we spoke about heterotrophic, and heterotrophic are going to be those organisms we call saprophytic nutrition. And saprophytic nutrition deals with the saprophyte, and the saprophytes are going to be your decomposers and detritivores. So your some bacteria, fungi, and of course, we are going to have like earthworms and things like that within that group. So that was some of what we covered last week. Then we hastened in last week's presentation to look at the equation for photosynthesis. And we said that there are 2 equations that the syllabus really want us to look at. They want us to look at the worded equation, and they also want us to look at the chemical equation. Now, the worded equation, what we said then is that water plus carbon dioxide combines in the process of photosynthesis, and the condition there, we said the water and the carbon dioxide, those would have been raw material, and the condition necessary for photosynthesis would have been chlorophyll, and of course you're going to have some enzyme there, and we're going to have the light energy from the sun. We did make some emphasis on, place some emphasis on light energy, because of course you'd understand that the sun has both light and heat energy. But for photosynthesis, of course, it's going to be the light energy. We continue to say, while that is the condition, we also have the product. And the product of photosynthesis is always going to be glucose and water. Then we looked at the chemical equation. And that equation is very, very important for us to understand. And I did promise that I'll be showing you how to balance the equation in one of our videos. Now, the chemical equation, you have to be very careful with this, more so for your multiple choice. While we said water in the worded equation, here we have to look at balancing the equation. So we are going to say 6CO2. So that is 6 molecules of carbon dioxide reacting with 6 molecules of water, that's H2O, and the condition remains the same. The condition is going to be sunlight and chlorophyll, and I'm going to come back there. Then we are going to have as product C6H12O6, and that's for glucose, plus 6O2. And 6O2 is the oxygen. But I really don't want to leave here as yet for these persons who are joining for the first time. I want to look at how is it that the plant get to use these things and where they are coming from. The fact that we're just coming from transport with active transport, diffusion, and osmosis, it might be the right time for us to talk about them. So let's go. So the plant is going to get the carbon dioxide from the atmosphere. And it is going to enter the plant via the stomata, a process referred to as diffusion. The plant is going to take the water from the soil in a process through the root here, of course, to the xylem vessel, a process referred to as osmosis. And then there are some ions that need to come from the soil, like magnesium, which is very important for photosynthesis, like iron, which is very important for photosynthesis, and of course, the nitrogen, which is very important for photosynthesis. These are going to be moving up to the plant by the process of active transport. Then we have to think about the products of photosynthesis, and of course, the oxygen from that is produced as a result of photosynthesis will leave the plant through the stomata in a process called diffusion. So there we have first a knowledge of diffusion, active transport, and osmosis. Very good. So we could move on. Then the syllabus wanted us to look at, for the bio students, we were supposed to look at the phases or the stages of photosynthesis. It is not so much required for human and social biology, but if you're a human and social biology student, it would bring no harm for you to understand. So there are two real phases or stages of photosynthesis, as it were, for the CSEC syllabus. There is the light dependent stage and the light independent stage. The light dependent stage and the light independent stage. I was just glancing at my notes now and I realized I should have made mention of this before moving into the light stage and into the light dependent stage and the light independent stage. It's very important for us to understand, where does photosynthesis take place? What part of the plant? Yes, you're going to see in the green part of the plant, more so the leaf. But if we were supposed to get to the cell, where in the cell, what organelle is responsible for photosynthesis? Can anybody tell me that? Of course, it's going to be your chloroplast. So the chloroplast is specially designed for that type of a thing. It will be trapping the sunlight and, of course, ensuring that the process of photosynthesis takes place. So let's up on into the stages or the phases of photosynthesis. Now, the first phase is going to be the light dependent phase. The first phase is going to be the light dependent phase. And what it means is that light is required here in order for photosynthesis to take place. Now, it is using the light energy. Please be reminded, for photosynthesis, it's always going to be the light energy. So there's somebody asking, what about if we have electricity? It's light energy. Doesn't really matter. And why we're saying that it is the sun's energy, because ultimately we think that all energy came from the sun. So we usually say it is energy from the sun because we're thinking whatever energy it is, it came pretty much from the sun. All right. So with the light energy in the first phase of photosynthesis, what happens is that the light energy from the sun is used to break the bonds of the water molecule that is taken up during that osmosis process, where it's taken up from the soil. Now, this water bond is being broken. Now, the oxygen from the bond will diffuse through the stomata of the leaf into the atmosphere. Then carbon dioxide from the atmosphere entering the plant leaf by the process of diffusion will combine with that oxygen in the light independent phase. So it is in the light independent phase that the hydrogen combines with carbon dioxide to make the glucose. Now, it's very important for us to understand here respiration versus photosynthesis. So one might ask, at what time does respiration take place? Now, respiration is always taking place. Organism is alive, respiration is always taking place. However, later in the lesson, we are going to show you that there are some limiting factors to photosynthesis that could slow or stop the process. So while we can have a stop on photosynthesis or a delay or that type of a thing, depending on the plant, we never have a stop for respiration until you're dead. Let's move on. So we looked at the conditions needed for photosynthesis. Then, of course, conditions, material, pretty much for this part here, we could put them in one group. So we could start by saying carbon dioxide. Just go to that formula that you have there, or the equation you have there. Carbon dioxide, water, light energy, chlorophyll. You're going to need adequate or the ideal temperature, because once there is a reaction within an organism, there is what we refer to as an enzyme. So you're going to have the right temperature in order for the enzyme to function. So we'll go with that again. We are going to need, start with your equation, carbon dioxide, water, sunlight, chlorophyll, ideal temperature, and enzymes. However, we understand that there will be no chlorophyll unless we have the following nutrient or the following minerals. We must think about magnesium. We must magnesium iron. We must think about iron, and we must also think about those nitrogenous compound that is needed for the process of photosynthesis and for the formation of that chlorophyll. We moved on down the line, and this is where the lesson will begin this week. I do hope that that recap was short and to the point, and there is something you can recall from that recap. If there's a question before I move on, please let me have it in the chat so that we can ensure that all is cleared up before we move on. Do we have a question? Is there a question that I need to answer, or do I need to ask a question? No question? That is strange. All right, I'm going to be asking you a question. I want you to name the 3 types of nutrition you need to know for the syllabus. As it's outlined by the syllabus, the 3 types of nutrition. All right, so name the 3 types of nutrition that is asked for on or by or within or the syllabus wants you to look at. Tell me the 3 types of nutrition very quickly. If you're following the notes, it shouldn't be so bad. We started out by talking about nutrition. So what are the 3 types of nutrition that they want us to make that distinction among? 3 types of nutrition. Very quickly, let me move on. All right, so if that is giving you a challenge, let me ask you another one. What's the site for photosynthesis? The site for photosynthesis. Are we having some problem with, let me just check my gadget here. I remember last week we had some problem. All right, this can't be. All right. So, no response. All right, I'll just remind you very quickly as we move along. Now, the 3 types of nutrition that the syllabus wants us to look at is going to be autotrophic nutrition, heterotrophic nutrition, and saprophytic nutrition. Now, the site for photosynthesis is always going to be the chloroplast in the cell. Now, we want to look at the adaptation of a leaf for photosynthesis, because the leaf is that chief area that is going to be capturing the sunlight for the process of photosynthesis. And there are many things that we want to look at. There are many adaptations. We want to look at about 10 of them today. Now, we're going to be doing them very, very quickly. And when I'm finished, if it's not raining on the outside or tomorrow, I want you to go outside and do some things for me. I'm going to be doing a little lab today. I'm going to be walking you through a lab as well. Now, I observe, before I get into the adaptation of a leaf, I realize that many students don't know what's a leaf. You ask them to get a leaf for a drawing lab, and they'll go and get a leaflet. So I'm going to be introducing you now to different types of leaves. Now, the mango tree, and I'm going to be calling some trees. In Jamaica, we have mango. I hope that's what it's called in other places in the Caribbean. The mango tree, it has a simple leaf. The mango tree has a simple leaf. All right, it is just one continuous leaf blade, and I'm going to try to do some quick drawing. So if we were supposed to look at the mango leaf, it would be something probably like that. I hope you can see. So it would be something more like this. The mango leaf, just a simple leaf like that. So that would have been what we refer to. It's a leaf that we would refer to as a simple leaf. All right, so the mango leaf, it doesn't really matter the shape. Once it's one continuous blade, it would have been a simple leaf. Very good. So that is the first type of leaf, and that is what most persons would expect a leaf to be. But what we find happening, if you know ackee, like in Jamaica, we have the ackee plant. I'm going to be drawing the leaf very quickly here. So here we have a petiole coming down. And then you might have something like that. Just give me a second. I'm drawing my leaf here. And this type of leaf for the ackee plant in Jamaica, is called a pinnately compound leaf. So what you'll find happening is that there is leaflets on both sides of the petiole. So there we have it looks more like an ackee leaf, and you are observing that there are leaves on both sides. So this particular leaf is a pinnately compound leaf. This leaf is a pinnately compound leaf. Now, if you were supposed to remove any one of these that you usually call leaf, that's just a leaflet. So I'm going to be circling a leaflet for you. All this here, all that area there. Let me use my crayon over here. I think I have some crayon over here. So I'm going to be circling a leaflet with my crayon. There we go. So one of these from the leaf is called a leaflet. All right. So this is from the ackee tree, and when you find something like this, you might not be paying much attention to leaves on the outside, but probably you could start today. Now, so that's the pinnately compound leaf, and then we spoke about the simple leaf. Now I'm going to be drawing the double pinnately compound leaf, and it's somewhat a hybrid leaf to the pinnately compound leaf. So very quickly, I'm going to be drawing that. Just stay with me. If you know the names of leaf, we could just pretty much put them on the screen, and we are going to be looking at them. So I'm not going to be drawing more. All right, so we are going to be having two on both sides here. I'll just draw a diagram rather than a drawing. And here we have another leaf coming up very quickly. All right, and it could go like that. So we could have one here. All right, so that type of a thing. All right, so the leaf is coming up very quickly for those persons who are not so certain what is a leaf. Usually we use a dichotomous key to identify the leaf. So this is a double pinnately compound leaf. Now I want you to look at this. All right, so this is the double pinnately compound leaf, and I'm going to be showing you the pinnately compound leaf. So let's go. This here is a pinnately compound leaf. See? See, so all this long area here would have been the petiole. All this here would have been the petiole. But what you have is that you have leaflet that radiates from both sides pretty much like a feather. All right? While with the double pinnately compound, it is somewhat different. Now, this is a double pinnately compound leaf, and this time we're having the petiole coming down the middle here. You see the petiole coming down the middle here, and the difference right here. So this is a double pinnately compound leaf. Now we're going to be showing you one more type of leaf, so that when you go outside to do your observation with leaves, it shouldn't be a big problem, because you would have at least known what a leaf looks like. So we're going to be showing you another leaf now. And this one you would find on like a flower we call alamander, and it radiates around a central portion of the petiole. It's like your hand palm, it's coming pretty much from one central point. So it would look something like this, right? And this one we refer to as a palmately compound leaf. So each, if we were supposed to remove any of the 5 here, we would have removed a leaflet. So this is a palmately compound leaf, and each of those things looking like a leaf, they are referred to actually as leaflet. Now, there's something else I need to bring you up to speed with. So we did look at some leaf, and I'm going to be showing you the leaf one more time before we get into leaf arrangement. So I'm going to be showing you first simple leaf. This is the one that everybody is accustomed to, and they might come in different shapes and all of that, but it's usually one continuous blade. Then after our simple leaf, I can show you our pinnately compound leaf. And this is our pinnately compound leaf. You'll observe that the leaflets are radiating from the petiole on both sides. So this here is our pinnately compound leaf. And then I could show you the double pinnately compound leaf. This here is the double pinnately compound leaf. And then we could continue and show you now what is called a palmately compound leaf. This is a palmately compound leaf. All right, very good. So what we're going to be looking at now, because we're looking at the adaptation of leaves for photosynthesis. And when you go outside to do this leaf lab, I'm sure you're going to be seeing different things and you might just be confused. So I hope that I've helped you just now with the leaf. Now, you probably would have seen a diagram of a leaf. And remember, when you're drawing, once you're drawing, you want to ensure that the labeled lines, please visit my video on diagrams of a leaf, or for drawing and diagrams, so you can have the rules as to how you should draw or not draw. It's right there. You could just go to my drawing video and you'll see that. Now, what I'm going to be showing you now, I'm going to be showing you leaf arrangement on the stem. This time not on the petiole, but on the stem. All right, so the first one I'm going to be showing you is this. You might see the leaves arranged on the stem, and they are one on one side and one on the other side. This type of leaf arrangement is called opposite. So they are opposite to each other. This leaf arrangement is called opposite. All right, this is an opposite leaf arrangement. I'm going to be showing you another type of leaf arrangement very, very quickly. Now, this type of leaf arrangement that I'm going to be showing you is one that we refer to as being alternate. All right, so it's more like a step. One is up and one is down. All right, so this is the alternate leaf arrangement. You observe one is down there and one is up there. So this is alternate, while this leaf arrangement is opposite. All right, so opposite, alternate. Opposite, alternate. Now, there's another type of leaf arrangement I'd like to show you very quickly before we move on. And this type of leaf arrangement is called the whorl. And what you find with the whorl, they are all connected at the same node of a stem, the very same node, but they are connected more like in a circle. All right, so they are connected right at the same place, pretty much in a circle. So this here would have been a whorl. And you would find this on, I think, the plant we call the alamander. The alamander would give you this type of leaf. So you see it's connected right around in a circle. Pretty much this is the whorl. So the leaf connection again, whorl. That's how the leaves are attached. This is a whorl. This is your opposite. And of course, this is going to be your alternate. All right, so we have looked at types of leaves. This is a very, very important lab we did just now. We have looked at types of leaf, and we have also looked at leaf arrangement. Now, before I get into adaptation, why not just take all the labs now? Please look in your textbook and be able to draw or describe the leaf. You must be able to identify the apex, the lamina, the veins, the petiole, the midrib, and of course, the margin. I'm going to be showing you another, I'm going to be showing you a lab. Just let me get the paper very quickly. All right, so I'll be doing a lot of drawing today. I am sure that you'll be enjoying what I'm doing. I'm going to be showing you a lab very, very quickly. For the students who are doing human and social biology, I think I said this in the last lesson, the syllabus wants you to be able to identify or to be able to carry out lab that shows that chlorophyll is needed for photosynthesis. They also want you to be able to prove that light is important for photosynthesis. Now, I am going to be using my markers, my crayons here to assist you with that process. Now, follow me very quickly. I'm going to be drawing a leaf very, very quickly. And what I'm going to be doing on this leaf, I'm going to be putting a paper strip over the leaf. So you're going to be seeing it in my diagram now. I'm going to be putting just a piece of paper over the leaf. And I'm going to, it's a block paper that I'm putting over the leaf, or a file, something to block out the sunlight. So it won't allow sunlight to touch this area of a leaf, right? Because we want to determine now if light is needed for photosynthesis. So you might have seen something like this on an exam paper. So this thing here blocking out the sunlight in this area. Now, we would have carried out the process of photosynthesis on this leaf. So if we were supposed to remove that black thing on the leaf, if we were supposed to remove it, then we'd still have this area here. And if we were supposed to test this now for starch, what do you think would happen? Remember here, no light was allowed to touch this area. No light was allowed to touch this area, right? And then we removed this black thing from over the leaf, and then we tested it for starch. Now, when we tested it for starch, what we realized, and I'm going to be putting up very quickly, I'm going to be using color here. Please don't use that in your SBA. I'm using it so that you can follow. So what we observe is that the blue color, the blue colored area here showed the present for starch. Now, that is the area changed to a blue-black color when iodine was added. However, this red area, I'm going to put it in red. This red area took on the red-brown color of iodine. All right? So let us go with this experiment again. Now, this experiment is to demonstrate that light is needed for photosynthesis. Now, we could do many things to prove that, but the popular experiment is to cover a portion of the leaf, as we have done here, cover a portion of the leaf, after which we are going to remove it probably within 2, 3, 4 hours a day, depending on what the lighting condition is like in your area. And then we are going to be testing the leaf for starch. Now, if the light was properly blocked out for long enough time, for a good enough period, what you will find happening is that the area that was covered will take the red-brown color of the iodine, while the rest of the leaf will take on a blue-black color. Now, the blue-black color is a color that signifies that starch is present. So this exercise here is to prove that light is needed for the process of photosynthesis. I'm going to be doing the other lab very quickly here for the persons who are doing HSB. The bio students need to know that lab as well, but I pointed it out for HSB because in most cases, HSB students are saying, we don't need to know in the lab, we don't need to know in the lab, but that is not true. There are about 10 to 14 labs, I don't remember quite well, in the back of your syllabus, Appendix 2 to be specific, that deals with labs for you. So I'm going to be doing what we call now a variegated leaf. Now, a variegated leaf is a leaf that has, sometimes it's yellow and green, sometimes it's white and green, but it's never usually one continuous color, as you would have found for most leaves. So we could look at this leaf here, and we could say that it is a variegated leaf, because what you find happening is that there are some areas of a leaf that is green and some areas of a leaf that is white. Now, the green area would contain chlorophyll. the white area would not contain chlorophyll. So when we would have tested this leaf for starch, what we would have found out is that the area that is green will turn blue-black, but the area that is white will take the red-brown color of the iodine. Now, guys, you need to know this. I know in most cases, the students who are doing human and social biology, you might not get an opportunity to do the lab. But to execute the lab, you could go to my video on testing leaf for starch. There's a video up on my channel. So go to my channel and look at testing leaf for starch. And you will see a variegated hibiscus leaf there. And you will see a fully green caronella leaf. All right. Watch the videos and make sure that you understand this. Remember, we're using iodine to do the starch test. So in the starch test, we're using iodine. And to remove the chlorophyll, we are using ethanol or alcohol. These are two wonderful experiments. I did talk about the other experiment in the last class. And of course, I'm going to be showing this family very, very quickly. So we would have had a beaker. I'm going to be drawing the beaker very quickly with a funnel. In the last class, I didn't do any drawing, but I'm doing very quick drawing now. Very, very quick drawing. In some cases, I'm going so fast, it's not so easy. So I'm doing that. I have no ruler where I am now. And then on top of this, we're going to be having a setup like this. Very good. And then we're going to be having that. Right? And I'm going to be putting in the water weed. Never mind, the lab is coming up very, very quickly right here. And it will help you, I am sure, in your exam. Now here we have some water weed. Pondweed, and what we want to do in this experiment is to prove that oxygen is given off during the process of photosynthesis. So I'm going to be using just some little pink color here to show the oxygen bubble going up and the gas accumulating at the top here. So here we go. I'm going to be showing it to you now. So here we have the setup. So you'll see a funnel that is turned over into a beaker with some water, and the water weed is that green looking thing at the bottom there. And then what you find, so here we have the water weed and the funnel is right here, and you're seeing a test tube turned over the funnel. Now, as the light, let me just draw in the light that you'll never ever forget that light is needed, whether it be sunlight or you're using a lamp, whichever one, it really doesn't matter. But usually there is light coming in. And this light will enhance the process of photosynthesis. So you'll see me drawing some rare light here coming from this lamp. And as such, as the process of photosynthesis takes place, you'll have some bubbles coming up, bubbles of oxygen, we hope, coming up, and it will be collected at the top of the tube here, and it will also displace the water that is in the tube. Now, this is just a part of the process. Remember now, we are testing in this experiment, we're trying to figure out if it is true that oxygen is produced during the process of photosynthesis. Now, the next thing that we're going to need to do, we're going to put our thumb over the test tube like that, and we're going to take it out all covered up, and then we're going to use what is called a splint, a glowing splint. Can you check to see if there's a splint in that thing for me, please? The craton over there with the equipment. All right, so you're going to be using a splint. It's something like a long matches stick, and then you are going to be putting it over a glowing splint. So you're going to light it and out it somewhat, so it will be like a destroyer just with that glow on it, not the burning flame. And then you are going to put it into the tube very, very quickly, or over the tube very, very quickly, and then you are going to remove your hand. Now, if you remove your hand and it relights the splint and start blazing again, then we know that oxygen was collected in the tube. Now, you must be very good at this in order for it to happen. If you're not good at it, probably you need to ask your teacher, or probably you need to just look on the internet for something, or try a couple of times, and you should be able to get over that. So I have done 3 labs so quickly. Time really fast, really fast. All right, so let us continue. We were looking at the adaptation of a leaf for photosynthesis, and I did show you some types of leaves. I did show you leaf arrangement. Then we did 3 labs. One of the lab we did was to check to see if light is needed for photosynthesis, and we use something to block the sunlight on the leaf, and we said that portion would have taken the red brown color of the iodine, and the part which was not color covered would have taken on a blue black color, which is the color, the positive test for starch. Then we also did another test where we were testing to see if chlorophyll is needed for the process of photosynthesis. So we chose a variegated leaf, a hibiscus leaf, of course, and we tested that. Now, the white area of a leaf is the area of a leaf that would be without chlorophyll, while the green area of a leaf would have had the chlorophyll. What we observe in that experiment is that the green area took on the blue-black color, which is a positive indication for the presence of starch, while the white area took the color of the iodine red-brown, which indicates that no starch, no chlorophyll was there. Chlorophyll is needed for the process of photosynthesis. Then we tried to find out if oxygen is given out during the process of photosynthesis, and I showed you the setup for that, and I told you what is expected of you. And once you follow all these processes, you will be fine. You'll be fine. The labs will be out in no time, and you'll be doing those on your past paper like poof. Now, if you want to see these labs as real lab and what they look like, visit my YouTube channel and look at the 3 past papers. I think I have 2013, 14, 2012, 13, and 14 past paper all work there. And all the experiments I did just now, they are on those 3 papers. Check them out and you'll see the papers are all worked and explained. You want to go on over and check those. Now, let us move on. I want to look at our leaf now. We want to look at the leaf and the adaptation of a leaf for photosynthesis. And I like to spell adaptation ADAP. That is the one that we are looking at. Adaptation. How is a leaf suitable? What makes the leaf suitable for the purpose of photosynthesis? And I said to you I was going to be looking at 9 things. I wanted to ensure that I kept the show in line, so I think I made some little jottings as to where I wanted to go with the show. I feel like I'm using so much paper, they're all over the place, but never mind. I could just treat you with the pictures that I have here. And the first thing I'm going to be telling you is that the leaves are flat. Can you relate to that? The leaves are flat for the purpose of photosynthesis. How does that help the process? Now, the leaves being flat, what they do, they increase the surface area. They are big, broad, and flat. They increase the surface area, hence it makes the leaf easier. It makes it easier for the leaf to be able to collect as much sunlight as quickly as possible. All right? Are you following me? Good. If you have any question, please share them. Please send them and I'll answer them right now. Let me have your questions and I'll answer them right away. Do we have any questions? No questions? No questions? All right. So the first thing we said is that the leaf is of course broad, flat. The other thing we want to say about the leaf is that the leaf is thin. So because the leaf is thin, it allows light to pass through easily. The other thing I'm going to be telling you about the leaf is that the leaves, they are located at 90 degrees to the sunlight. So what it means, you're looking at a leaf, and of course, let me look at the leaf. So the leaf, I drew several leaves here. All right, so we're looking at a leaf, and you will observe that at all time, the leaf will turn itself to ensure that it is getting as much sunlight as possible. You might want to look at a runner, one of those plants that like a vine that is running on a wall. And it doesn't matter which way the runner turns. The leaf is always will be angled towards the sunlight. You want to take note of that. So we said that the leaf is flat and broad. That increases the surface area for collecting light. We said that the leaf is thin, so it allows the light to pass through very quickly. And again, we said that the leaf, they are at 90 degrees so that they capture as much sunlight as possible. Now, the arrangement, the leaves are so arranged, and I show you the arrangement just now, depending on the plant. There are some leaves that are attached in a whorl format to catch as much sunlight. There are some that are attached as alternate, and there are some that are attached as opposite. All these enhances the process of photosynthesis in these plants. Now, another thing I want to talk about, we could get into the leaf. There's a big network of veins, right? Veins that are carrying the nutrient, carrying the water, pretty much moving things around the leaf. The other thing we could talk about is that waxy cuticle that is on the upper layer of the leaf, that despite the leaf is carrying out its many processes, the plant is not losing more water than it should lose. Now, another thing we could talk about is the stomatal pores, which are located under the bottom of the leaf, and they would enhance the process of gaseous exchange. Now, we could talk about the location of the palisade mesophyll. The palisade mesophyll, it is located closer to the upper surface of the leaf, and that is where we are having a whole lot of the whole photosynthesis process taking place in the palisade mesophyll. But we don't want to leave out the spongy mesophyll, because the spongy mesophyll is so well spaced that it allows for the movement, the easy movement of water and gases within the leaf. Now, never ever forget the xylem vessel and the phloem vessel. And I'm going to be doing a quick drawing of that as well. I hate to talk about these vessels and not show examples of them, because I know these are things that are always coming up. More so, this is for the bio students. They are always coming up in your bio exam. Remember that the xylem vessel, they are dead. And of course, they are just reduced to what we refer to as some hollow tube. So this here would have been your xylem vessel. This here would have been your xylem vessel. Usually it is like that. But you could see it on a paper just drawn as a circle. All right? You could see it looking at the internal structure of a leaf. It could be drawn as a circle. So if the xylem is drawn as a circle, then the question is, what would the phloem look like? The phloem is usually drawn, and I'm going to be drawing it right now. It's usually drawn with a companion cell to the side, and you will have just some little dots inside, might be 2 or more. And why you have 2 or more, this here is the phloem. Why you'll have 2 or more dots in it? Because the phloem has sieve tube. The phloem is alive. So we said that the xylem was dead. The phloem is alive, and to the side of a phloem, we're having the companion cell. Now let me draw the phloem in another view. All right, so you're going to be having the companion cell to the side here, and of course you're going to be having a sieve tube. It's not like the xylem vessel. So here you'll see the phloem cell. It has a companion cell, and the companion cell has a nucleus. So it looks totally different than does the xylem vessel. But remember, the xylem vessels are very important in transporting water and mineral salts from the soil to other parts of the plant, while the phloem vessel would be transporting the manufactured food from the leaf and the other green areas of the plant to other areas in the plant where it's needed. All right, so remember that the phloem is transporting food, while the xylem is transporting water and mineral salts. And those are very, very important adaptation that the leaf has, because without these, then the leaf would not be able to effectively carry out the process of photosynthesis. Now, that's a whole lot said. That's a mouthful. How much can we recall? So let's go through them very quickly. And I want to go through them where you can visualize it. So we are looking at outside now. The first thing we know is that most leaves, they are really broad. They are broad and they are flat. The other thing that we know about leaf is that they are thin, and that allows sunlight to go through very quickly. We also know that leaves lie at 90 degrees to that stem in order to capture as much sunlight. We know that we have the palisade mesophyll, which is closer to the upper surface of a leaf, and it is where we're carrying out the bulk of the photosynthesis process. We spoke about the mesophyll layer, which is wildly spaced to allow that diffusion of gases and the movement of water within the leaf. We spoke about leaf arrangement, and we spoke about three types of leaf arrangement. We spoke about opposite, we spoke about alternate, and we spoke about whorl, WHORL. We spoke about whorl. We also spoke about the waxy cuticle, which protects the leaf from the excessive water loss. And we did talk about the xylem, phloem, and of course, we spoke about the stomatal pores and the veins in the leaf. That's a lot said. That's a mouthful. I observe that no one is talking this week. I'm wondering if we're having the same problem where we had last week. But the show will go on. You could just send me the message and I'll look at the video and I will respond next week. But we're going to be going through with this show right to the end. All right, let us go. We're doing well so far. Now, we want to now look at the factors. The factors that's affecting the rate of photosynthesis. We did that already. Factors affecting the rate of photosynthesis. Did we do that? All right, very good. Let's go. No, we didn't. What we looked at was the condition necessary for photosynthesis. Now, the factors which are affecting the rate of photosynthesis is going to be light, temperature, water, and carbon dioxide. Let's go again. The factors, don't mix them up now. We spoke about condition needed for photosynthesis. Now there are four factors. Four factors that will affect the rate of photosynthesis. These are the factors. It's okay. I am listening, sir. Very good. All right. So there are four factors that will or can affect the rate of photosynthesis. And these four factors we made mention of: light. If we don't have adequate light, the process of photosynthesis will be affected. And in that case, we're going to be calling light a limiting factor. But before we move on, I want to ensure that I give you a wonderful lesson. I want to make reference to the fern in Fern Gully. The fern in Fern Gully. The thing about fern, in most cases, they are found on the forest floor. As such, they are exposed to but little sunlight. In order to make up for that, the fern will produce more chlorophyll. In producing more chlorophyll, it will capture as much sunlight as is needed for its whole process. Now, let's continue. Now, so this light could be a limiting factor. And of course, at night, you know that for the CXC level, I like to talk about the CXC level because pretty soon you might just call me back and say, sir, you never tell me about no COM plant, or you never tell me about no C4 plant. I really don't want to go there. I don't want to take on that at this level. It is not on your syllabus, so I won't touch it. When you go to Cape, we will look at that. Davika Samuels, big up yourself. How are you? Keep up the good work, sir. Sir, I am so proud of you. This is one of my students who pretty much at the end of the line, I told him, I have to stop the lesson to talk about this. Very, very good. This fellow was a fellow I thought that had the potential and was not giving me a pound for pound. And I spoke to him a couple of days close to the exam. And trust me, he came out victorious. Of course, he didn't get a 3. His grade is better than a 3. I am proud of you, son. I hope you're out there doing some good work. We are continuing the story of the factors that are affecting the rate of photosynthesis: light, temperature, water, carbon dioxide. Now, temperature is going to be a limiting factor. And let me tell you something. Anywhere that you have enzymes working, temperature will always be important. I do hope that what I'm going to say now will not hurt anyone, but it is usually something I make reference to. There was a sad situation somewhere in Mountain View, some gas fire thing like that. And there were persons who made it out, but later we had sad story. Now, the thing about it, once the body temperature would have gone way above optimum, 37 thereabout, way above that, You start to go in 50 and 60. You are being cooked alive. Even if you are able to talk after that ordeal, the chances of you living will be but little. So let us go. If we have very cold condition, the enzymes will not work. So that is going to be a limiting factor and the process will not take place. If we have very, very high temperature, you have seen it when they do slash and burn, and it's not the plant that you see dry up at the side that had the fire. It was just exposed to this high temperature. That is a limiting factor and could cause the death of the plant. Now, water, oh my works. You would have heard the farmers complaining. The drought, the government is not giving us any water. The rain is not falling. Oh my works. When there is no rain, as it were for small farmers and subsistence farmers, that affects them greatly. And of course, the plants will bear but very small fruit, and some won't bear any at all. So water is a very important part of photosynthesis. And if you're an agriculture student, you must have mastery of photosynthesis because you have to understand how it works. You're not going to be planting some cucumber underneath a well-shaded tree and you get a lot from it, because of course that's going light is going to be a limiting factor there in getting the plants to bear. Now, carbon dioxide, there's just about 0.04% of carbon dioxide in the atmosphere. However, if the amount of carbon dioxide is increased, probably closer to 1, it increases the rate of photosynthesis. So always remember, there are really 4 limiting factors for photosynthesis. We discussed them here today. We spoke about light intensity. We spoke about temperature. We spoke about water. And we spoke about carbon dioxide. Always remember that plants are what we consider to be carbon sinks. Let us continue. We're going to be looking at the last thing for this lesson. And I will tell you what's coming up on Sunday and what we have planned for next week. So we're going to be looking at the last thing. Now, you would have heard this or you would have seen this on the syllabus. The syllabus is asking for both HSB and biology. What's the fate of products produced in photosynthesis? What's the fate? What it means is what happened to them. The fate of the product produced during photosynthesis, all it means is really what happens here. All right, Shakila Lewis, big up yourself. All right, so the fate of the product, we're going to be looking at both. So I want to look at oxygen first. Now, oxygen is produced by the plant. And of course, as animals, we use oxygen. The question is, plants use oxygen? Yeah, they do. If a plant is carrying out respiration, then it must use oxygen because aerobic respiration is going to require some oxygen. So yeah, the plant is using some of the oxygen while it is giving off some. Morrison, big up yourself. Tanaya Morrison, enough respect to you guys. I'm very happy that you're all here. All right, good. So the plant is using some oxygen for respiration. It is giving off most of that oxygen to the atmosphere. You and I, all animals, all processes that require oxygen for aerobic respiration, they are using this oxygen for respiration. But also processes like combustion, burning, when you're cooking the stove, like the stove, it is using some oxygen too for combustion, burning. Yeah, combustion is a type of respiration, but the thing about combustion is that it produces way more energy. All right? So yes, oxygen is used for combustion, oxygen is used for respiration, and I'm sure there are many other stuff that oxygen is used for, but those are the two closest to my mind. Now, starch. The glucose that is produced, some is converted to starch, some is converted to protein, vitamins, some used to make chlorophyll, and the plant uses some for its daily function. Now, we also consume some of that glucose that is converted and stored in starch. So, of course, we are able to survive. Now, Santina Poisa, big up yourself. I'm very happy to see all these bio fans. I tell you, I was told that at one school, you know, next week is going to be more. Big up yourself, Angel Moore. No, I was told that at one school, next week is going to be your monthly test. No, I don't want anybody to be failing the monthly test, you know. So I am going to say to you guys, please ensure that you're up on over to CSEC Biology, the cover page, or you could type in Mr. Wilson, CSEC Biology with Mr. Wilson, or Mr. Wilson. Type in those things at YouTube. I mean, you can type in almost everything to find me at YouTube. I deliberately did not put up much more video this week because in most cases, most person would have covered just what we covered so far on the syllabus because we've been going pretty fast. So that brings us pretty much to the end of the delivery for today. There is just one thing we have left to look at on photosynthesis, and that is going to be looking at the plant nutrient. We have made mention of nitrogen, magnesium, and iron. In our lesson next Friday, we are going to be taking on this as a lesson by itself. We are going to be looking at calcium, we're going to be looking at phosphorus, we are going to be looking at sulfur. We're going to be taking on these by themselves because what I know as a fact is that in most cases, the plant nutrient, they end up in the null curriculum. Yeah. And we don't want to get it there. So here we're going to be exploring just about everything. So tell you what, we have been looking at circulation at grade 11. I don't know where the rest of my fans are, the rest of my viewers, my subscribers. But come this Sunday, I'll be looking at circulation. Now, for those persons who do not understand the concept of surface area to volume ratio, I have done a video to explain how we arrive at it mathematically. Now, I have another video that is coming out. Well, it's finished. It should be posted or uploaded this evening. I have another video that will now show you the biological side. How do we apply the surface area to volume ratio to biology? So remember, we have another show coming up on Sunday at 5. We try to make the show just 1 hour because we know you want to enjoy your weekend. You want to have your chicken and fries, your hot wings, your jerk chicken neck, and the list of stuff goes on and on and on. Now, I'm very, very happy to have you here. I want to shout out to all my fans in Jamaica. Big up yourself. I want to shout out to my fans in Trinidad and Tobago. Big up yourself. We're seeing you as well. I have some fans over there in Guyana. I want to say hi to you as well. We have some persons who are linking us from India. We have to shout them as well. We have some fans coming to us from the United States of America. We want to say enough respect to you in the Jamaican way. And those from England, we want to say enough respect to you in the Jamaican way. Enough love, one love, whichever the love you want to take from Jamaica, it's just pure love. I do hope that we are doing all our best to make it to the A spot. To make it to the A spot. Yes, Creek, I'm seeing you. Big up yourself. Creek was the first person on this evening. Now, let me tell you one thing before I go. I need 13 subscribers this evening. 13, 5, 5, 3, to make it to 500 subscribers in 2 months. I am so proud of you all. Nitisha Francis, big up yourself. I am so proud of you all. You helped me to, I mean, I'm getting some things from YouTube that is looking like really wonderful. We're not monetized as yet. As soon as I am monetized, you will know because my output will be better. My video quality will be better. I'll be purchasing better software. So I want you to help me this evening to reach my 500 subscribers in 2 months. We're doing well. 500 subscribers in 2 months. Big up all those schools in Jamaica. I am seeing you. Enough respect. Big up to the parents. I didn't say much about the parents. And there are some church groups that are a part of the family. I want to big you up as well. And there are some other civic groups that are a part of the family. Remember, even if I am not your teacher, even if you don't know me, it is fine to join the family. My name is Wilson. They call me Mr. Wilson. Feel free to join. And if you don't know my name, to say sir or whatever way you refer to me as, that is fine. I'm very, very happy to have you this evening. I'm going to be leaving you now, but remember, at 5 p.m. on Sunday, we go circulation, and I'll be cutting up that art left, right, and center. I think today we had a square art with some students where we really wanted them to understand all parts of the art, and we're going to be looking at that. We're going to be looking at why multicellular organism need to have a complex circulatory system. We're going to our transport system. We're going to be looking at what is it that has been transported by the circulatory system. We're going to be we're going to be describing the heart and talk about its function. We're going to be looking at blood vessels that supply specific organs. And of course, we're going to be looking at the blood vessel and their makeup. We're going to be talking about the vein. A lot of persons, I made mention of the varicose vein. That's swollen vein when the valve in the vein is malfunctioning. We made mention of that, and we're going to be looking at that. I want you guys to be doing well. So if you find that you're having problems, just send me a comment with one of the video. Please ensure that you're watching the videos to the end. Please ensure that you're inviting your friends over to subscribe, like, and share. This is a big family, and we meet here every Friday at 5 p.m. right up until exam. And of course, on a Sunday, we are going to be meeting over there at grade 11, HSB and Biology, where we are going to be looking at, for this week, we're going to be looking at circulation. So I'll be covering grade 10 on a Friday, and grade 10, you can join me again with grade 11 on a Sunday. And grade 11, you can join me on a Friday and that type of thing. You understand what I'm saying? That's where I'm going. Peace out. Thank you very much for joining. Love. Have a wonderful weekend. Stay in, stay away from flooded areas. Stay away from COVID. Remember to sanitize. Remember to wear your mask. Remember all of those fancy things that you are supposed to do, because I want to see you here on Sunday, and I really don't want to have any sad story of any of you. I'm losing any of you. All right. Love, peace, thanks. Say I, and thanks to your parents for providing the resource for you to be logged on. Bye-bye.